Okay, let's unpack this. Have you ever marveled at how your body keeps thousands, literally thousands, of complex processes humming along perfectly? Adjusting to everything from that big meal you just ate to an intense sprint at the gym.
Yeah, it's pretty amazing. It's not magic, obviously. It's all thanks to this incredibly sophisticated control system for the tiny cellular workhorses we call enzymes.
Right, the enzymes, the
But
workhorses.
exactly. Think of them as like the essential machinery in your cells. But like any good factory, they need really precise regulation. So our mission in this deep dive really is to explore exactly how these diverse enzymes are controlled, making sure they perform their vital functions efficiently, without waste, and always adapting to your physiological state, your diet, the environment.
All of it.
All of it. We'll try to uncover the fundamental mechanisms and, frankly, the ingenious strategies that orchestrate the metabolic pathways they power. Basically, summarizing what you'd find in a resource like Marks Basic Medical Biochemistry.
And we're not just staying theoretical. We'll connect these intricate controls to real-life scenarios, like understanding maybe why someone struggles with alcohol metabolism or how an athlete like Ann R. in the case studies fuels her muscles mid-jog.
Yeah, it's truly a dance of biological precision. We'll describe these examples so you can picture them. #1, the fundamental principles of enzyme control.
Okay, so when we talk enzyme regulation, where do we even begin? It seems so complex.
Well, we always start with a core idea, a fundamental principle from the chapter. Regulation is perfectly tailored to an enzyme-specific job. In any metabolic pathway, a series of chemical reactions, there's almost always a bottleneck
It's
enzyme.
a bottleneck. Okay.
Yeah, this is the regulatory enzyme. It's the one that catalyzes or controls the slowest step, the rate-limiting step. By fine-tuning this single enzyme, the body can adjust the speed of the whole pathway.
Just controlling that one choke point.
Exactly. And it's not just about turning existing enzymes on or off faster or slower. The body also controls the actual amount of enzyme present. So it can make more enzyme protein or break some down. That changes the pathway's maximum capacity over the longer term.
Right. So both speed control and, capacity control.
Precisely.
Now, it feels intuitive that the amount of raw material, the substrate, would affect how fast an enzyme works. What's the science behind that, the basics?
You've hit on a really crucial point. All enzymes show what we call saturation kinetics. Imagine a tiny factory assembly line. If you only give it a few parts, it works kind of slowly.
Makes sense.
Give it more parts, it speeds up. But eventually, right, the line is running at its absolute maximum capacity. All the workers are busy. Adding even more parts won't make it go any faster. That's saturation.
Okay.
And that maximum speed, that's what biochemists call Vmax, maximum velocity.
Vmax, got it. And how do we measure an enzyme's, eagerness for those parts? It's hunger.
Good question. That's where a key term from the Michaelis Menten model comes in. Km. Think of Km as a measure of an enzyme's affinity, its hunger, for its substrate, the molecule it acts on. A lower Km means the enzyme is super efficient. It gets to half its maximum speed, half Vmax, even when there's very little substrate around.
So it's really good at grabbing what it needs, even if supplies are low.
Exactly, like a hungry worker grabbing the first few parts. This property is absolutely crucial in how different tissues handle the same molecule, say glucose, depending on their unique needs.
Can you give us an example, something relatable where this Killam difference really matters?
Absolutely. Let's consider how your body handles glucose, like the chapter describes. Your red blood cells, for instance. They're always working, always needing glucose fuel, right?
Right, constant need.
Even if your blood sugar levels dip a bit low. So they have an enzyme called hexokinase I1. This enzyme has a very, very low kilomemmer for glucose, about 0.05 millimolar.
Tiny number.
Super low. It means it's incredibly efficient at grabbing glucose, basically working near its Vmax almost all the time, keeping those crucial cells fueled. Now, compare that to your liver. The liver's job isn't just to use glucose immediately. It also needs to store any excess after a meal.
Yeah, different role.
Totally different role. So the liver has a different version of the enzyme, an isozyme called glucokinase. And guess what? It has a much higher kilomeme for glucose, around 5 or 6 millimolar.
Much higher, yeah.
Which means liver glucokinase activity only really ramps up significantly when blood glucose is high, like after you've eaten a lot of carbs. It's perfectly designed to promote glucose storage as glycogen or fat only when there's plenty around.
So like in the case study, Ann R. eats a high-carb meal.
Exactly. After that meal, her liver glucokinase springs into action. It goes from maybe 44% of Vmax at normal glucose levels to, say, 80% of Vmax when glucose is high. really working hard to process all that incoming sugar.
That's a fantastic illustration. Really shows how regulation matches function. Different tools for different jobs.
Precise.
What about a more complex case, like alcohol metabolism? How does Curran play out there, maybe with LM's situation?
Right. Alam's case in the text highlights this well. With alcohol metabolism in the liver, there are basically two main enzyme systems involved. First, there's liver alcohol dehydrogenase, or ADH. This one has a very low kin lemma for ethanol, about 0.04 millimolar.
So, very efficient, like hexokinase.
Yeah, highly efficient, which means it gets saturated really quickly, even at relatively low blood alcohol levels, often below the legal driving limit. It tries its best to clear that alcohol fast.
Okay, so it maxes out easily.
It does. But then when ethanol levels get high, like after several drinks, a backup system comes into play. This is sometimes called MEOS, the microsomal ethanol oxidizing system.
Right.
And this system has a much, much higher kilomass, around 11 millimolar.
So it only really gets going when there's a lot of alcohol.
Exactly. It becomes much more active at higher concentrations. Now, while this backup system helps clear the excess ethanol, which is good in the short term,
There's a downside.
there's a potential downside, yeah. Its byproducts, unfortunately, can contribute to liver damage over time, possibly leading to things like cirrhosis if alcohol consumption is chronic. It's like a trade-off the body makes.
Wow. So the body's juggling these different systems, different M's depending on the situation.
Absolutely.
But it's not always about speed, right? Sometimes the goal is to slow things down, put the brakes on. How do enzymes get inhibited?
That's another crucial part of regulation. Enzymes can be reversibly inhibited, meaning slowed down temporarily, by compounds that bind to them. We usually classify these inhibitors based on how they interact. A common type is the competitive inhibitor.
Competitive. Like they compete for something.
Exactly. Often, it's a molecule that looks structurally very similar to the enzyme's normal substrate. It literally competes for the active site. That's the spot where the reaction usually happens.
Okay.
Now, the cool thing is, if you flood the system with enough of the real natural substrate, you can often overcome this type of inhibition. You basically outcompete the inhibitor.
So you can push the decoy out of the way?
Kind of, yeah. It makes the enzyme appear less hungry for its real substrate, so it increases the apparent key emitter. But importantly, it doesn't change the enzyme's maximum potential speed, the Vmax, if you get enough substrate in there.
Interesting. So it raises the chemo, but leaves Vmax alone.
Right. Now, contrast that with the non-competitive inhibitor. This one's different. It binds to the enzyme, but not at the active site. It binds somewhere else.
Okay, a different location.
Yeah, and this binding changes the enzyme's overall shape, which then reduces its effectiveness. It's like it subtly damages the enzyme's machinery. It essentially takes some of the enzyme out of commission, effectively lowering the Vmax, the maximum speed. And crucially, adding more substrate won't overcome this type of inhibition. It doesn't change the Km.
So non-competitive lowers Vmax. Competitive raises apparent Kmax. Got it.
Are there medical uses for this, like competitive inhibition?
Oh, absolutely. A classic life-saving example is treating methanol poisoning.
Yeah.
Methanol, you find it in things like windshield wiper fluid, is incredibly toxic when your body metabolizes it via alcohol dehydrogenase, ADH.
But ethanol, the alcohol we drink, is structurally similar to methanol. So doctors can give a patient suffering from ethanol poisoning controlled doses of ethanol. The ethanol acts as a competitive inhibitor. It competes with the methanol for the ADH active site.
Buys time.
Exactly. It dramatically slows down the conversion of methanol to its toxic byproducts, like formaldehyde, giving the body a chance to clear the methanol safely. It's a really neat clinical application.
Wow, that's clever biochemistry in action.
Yeah.
What about the products of reactions? Can they slow down the enzyme that just made them?
They certainly can, and often do. It's a fundamental concept.
Yeah.
All products are actually reversible inhibitors of the very enzymes that produce them. We call it simple product inhibition.
Simple product inhibition. Okay.
And it's vital. It prevents one enzyme in a pathway from, like, racing ahead and overwhelming the next enzyme with too much product. For instance, glucose 6-phosphate, the first product in glucose breakdown, inhibits hexokinase, the enzyme that makes it.
Makes sense. Don't make more if you already have plenty.
Exactly. It ensures that if the cell already has enough of this G6P, new glucose isn't immediately snatched from the blood, leaving it available for other tissues that might need it more urgently. And looping back to LM and alcohol,
Yeah.
the NADH that's generated during ethanol oxidation by ADH, that NADH actually acts as a product inhibitor for ADH itself.
So it slows down its own breakdown.
To some extent, yes, it contributes to slowing ethanol clearance, and that buildup of NADH can also cause other problems, like inhibiting fatty acid breakdown, which can contribute to fatty liver disease. It's all interconnected.
Okay, this is where, for me, it gets really interesting. Enzymes aren't just static things waiting for substrates or inhibitors. They can actually change their shape, right? Conformational changes.
Oh, absolutely. You've hit on a core truth emphasized in biochemistry. Enzymes are dynamic. These conformational changes are incredibly powerful regulatory mechanisms, often happening far from the actual active site. It includes fascinating strategies like allosteric control, covalent modifications like adding chemical tags, and even dynamic protein-protein interactions. A. Allosteric enzymes. Remote control.
Allosteric. That sounds pretty scientific. What does it actually mean for how an enzyme works?
Think of it like having a remote control for your enzyme. Allosteric means other site. So allosteric activators or inhibitors, we call them effectors, don't bind at the enzyme's active site where the main action happens. They bind at a different, distinct site on the enzyme.
A regulatory site.
Exactly, a regulatory site. And this binding, even though it's remote, causes a change in the enzyme's overall 3D shape. A conformational change. This shape change then subtly, but powerfully, alters the active site's ability to bind its substrate or catalyze the reaction, often making it better or worse. And many allosteric enzymes, especially the key regulatory ones, are often made up of multiple subunit, multiple protein chains working together. They frequently show something called positive cooperativity.
Cooperativity.
Yeah, it means that once one subunit binds its substrate, it actually makes it easier for the other subunits to bind their substrates. It's like the first binding event sends a signal through the enzyme saying, okay, guys, let's get to work.
So one signal can create this chain reaction within the enzyme, boosting its efficiency.
Precisely. It leads to a much sharper response to changes in substrate concentration. And this also means if you plot the enzyme's activity, its velocity, against the substrate concentration, you often don't get that simple hyperbolic curve like with Michaelis-Menten enzymes. Instead, you often see a sigmoidal curve, like an S shape.
An S-curve, okay.
And allosteric activators typically shift this S curve to the left. What that means functionally is they decrease the enzyme's apparent corratum, making it much more sensitive and responsive to its substrate, even at lower concentrations.
So it turns on more easily.
Right. And inhibitors, they do the opposite. They shift the curve to the right, increasing the apparent telomere, making the enzyme less sensitive. It needs more substrate to get going.
Why is this S-curve, this allosteric type of regulation, so vital? Why not just stick with the simple model?
The genius of allosteric regulation is its ability to provide almost instantaneous and sometimes massive adjustments to enzyme activity. It acts like a cellular dimmer switch or volume knob, not just on-off. It allows for these exquisitely sensitive and rapid adjustments based on the cell's immediate needs, like sensing energy levels.
Energy levels.
Yeah. For example, when your muscle cells are working hard and running low on ATP, the energy currency levels of ADP and AMP rise. These molecules, ADP and AMP, act as crucial allosteric activators for key enzymes in energy-producing pathways, like glycogen breakdown or glycolysis.
Signaling the need for more ATP.
Exactly. They rapidly switch on those pathways. It's incredibly fast and potent, much faster than, say, making more enzyme protein. B, covalent modification, adding a molecular switch.
Okay, so beyond just things binding to the enzyme, you mentioned enzymes can actually be chemically modified, like adding a molecular switch.
Yes, exactly. That's another major category of regulation, covalent modification. And arguably the most common and important type is phosphorylation.
Phosphorylation, adding a phosphate group.
Precisely. Specific enzymes called protein kinases take a phosphate group, usually from ATP, and chemically attach it, covalently bond it, to certain amino acid building blocks within the target enzyme, typically serine, threonine, or tyrosine residues.
Okay.
Now, this added phosphate group isn't trivial. It's bulky, and it carries a significant negative charge. So adding it causes a pretty dramatic change in the enzyme shape, its conformation.
And that shape change is the switch.
That's the switch. It can flip the enzyme from an inactive state to an active one, or vice versa, depending on the specific enzyme. And importantly, this is reversible. Other enzymes, called protein phosphatases, can come along and snip that phosphate group back off, reversing the change, turning the switch off again.
So you have kinases adding phosphates and phosphatases removing them. A dynamic balance.
A very dynamic balance, often controlled by upstream signals like hormones.
How does this phosphorylation switch play out in that real-life scenario? And our jogging.
Right. Let's go back to Anna jogging, like in the case study. Her muscle glycogen phosphorylase, that's the enzyme that breaks down stored glycogen into glucose for energy.
The fuel release enzyme.
Exactly. It isn't just activated by those allosteric signals like A and P we just talked about. It's also very powerfully activated by phosphorylation. This phosphorylation is done by another enzyme called glycogen phosphorylase kinase. Now, stay with me here.
Okay.
When Anne's body releases adrenaline, epinephrine, during exercise, that hormone triggers a whole signaling cascade inside the muscle cells. This cascade leads to the activation of glycogen phosphorylase kinase, which then phosphorylates and activates glycogen phosphorylase itself.
So it's a chain reaction.
It's a chain reaction, yes. And this ensures a rapid and sustained breakdown of glycogen, providing fuel for her working muscles, even if maybe her immediate AMP levels haven't changed drastically yet or start to recover. The hormonal signal overrides or adds to the local signals.
You said cascade.
Yeah.
That sounds like it could amplify the signal.
It absolutely does. That's a key feature. Think about it. One hormone molecule outside the cell can lead to the activation of, say, protein kinase A inside. Protein kinase A is often activated by a second messenger molecule like cyclic AMP or CAMP, which itself is produced in response to the hormone.
Okay, hormone protein kinase A.
Right. Then, one active protein kinase, a molecule, can phosphorylate and activate many molecules of the next enzyme in the cascade, like glycogen phosphorylase kinase.
Amplification.
Exactly. And then each of those activated glycogen phosphorylase kinase molecules can phosphorylate and activate many molecules of the final target, glycogen phosphorylase.
Wow, so a tiny initial signal gets massively boosted.
Massively amplified. This allows a small hormonal signal to have a huge coordinated effect on many different metabolic pathways simultaneously, getting the whole body ready for fight or flight or sustained exercise. C, protein interactions, dynamic partnerships.
So beyond binding small molecules or adding phosphates, enzymes can actually team up, form partnerships with other proteins to change their activity.
Absolutely. This is another layer of regulation. Sometimes specific modulator proteins bind to enzymes. This binding can induce conformational changes, similar to allosteric effectors, or they might even physically block the enzyme's active site.
Okay. Any key examples of these modulator proteins?
A really important one, especially in muscle, is a protein called calmodulin. Calmodulin binds calcium ions, Ca2 plus.
Calcium, right? Involved in muscle contraction.
Exactly. So during muscle contraction, when a nerve impulse triggers the release of calcium ions inside the muscle cell, these calcium ions bind to calmodulin. The resulting Ca2 plus calmodulin complex then binds to and activates that enzyme we just mentioned, glycogen phosphorylase kinase.
Linking contraction directly to fuel mobilization.
Precisely. It ensures that the energy supply system, glycogen breakdown, is rapidly ramped up exactly when the muscle needs the energy for contraction. It's a beautiful link between signal and function.
Very neat. And what about G proteins? We hear that term a lot in cell signaling. Are they involved here too?
Yes, G proteins, particularly the smaller monomeric G proteins like RAS, are fantastic examples of regulation through protein interaction. They act like molecular switches.
Switches again?
Yeah, they cycle between two states, an on or active state when they are bound to a molecule called GTP, and an off or inactive state when they're bound to GDP.
GTP on, GDP off.
Right. When GTP is bound, the G protein changes its shape, its conformation. This new shape allows it to bind to and activate or sometimes inhibit a target protein, maybe an enzyme or another signaling molecule. And they even have a kind of internal timer. They slowly hydrolyze or break down the bound GTP into GDP.
Turning themselves off.
Turning themselves off, exactly. This makes the signal transient. This precise on-off cycling, which is itself often regulated by other accessory proteins, is absolutely critical for controlling countless cell processes. Everything from cell growth and division to how cells move around, deproteolytic cleavage, an irreversible switch.
All these mechanisms seem reversible. Inhibitors come off, phosphates get removed, proteins unbind. But are there ways to activate an enzyme, like permanently, an irreversible switch?
Yes, there is. That mechanism is proteolytic cleavage. Proteolytics means protein cutting. Some enzymes are actually synthesized and stored in an inactive precursor form. These precursors are often called zymogens or proenzymes. Think of them as dormant enzymes safely stored.
Zymogen.
Then, to activate them, a specific part of the precursor protein chain is snipped off by another enzyme, a protease. This cleavage event causes a permanent conformational change that reveals the active site or properly forms it.
And it's irreversible. Once cut, it's active.
Generally, yes. Once that piece is cleaved off, the enzyme is active, and it usually stays active. There's no going back to the zymogen form.
Why use such a permanent method? Seems risky.
It's used in situations where you need activity to be strictly localized or primed, often to prevent damage. Think about digestive enzymes like trypsin or chymotrypsin. They're made in the pancreas as inactive zymogens, trypsinogen, chymotrypsinogen. If they were active in the pancreas, they'd digest the pancreas itself.
Oh, right. Bad news.
Very bad news. So they're only activated by proteolytic cleavage once they safely reach the small intestine where they're needed to digest food. Similarly, many proteins involved in the blood clotting cascade circulate as inactive zymogens like prothrombin and fibrinogen. They only get cleaved and activated to thrombin and fibrin right at the site of a blood vessel injury, allowing a clot to form precisely where it's needed, not just anywhere in the bloodstream. It's a critical safety mechanism.
Okay, we've covered a lot of ground on individual enzyme controls, Km inhibitors, allosteric, phosphorylation, cleavage. But how do these all weave together to regulate entire metabolic pathways? The bigger picture.
Yeah, that's the key question. How does the cell orchestrate all this? The overarching principle, as emphasized in texts like Marks, remains the same. Regulation perfectly matches the pathway's overall function in the body, and typically pathways are regulated most strongly at their slowest step, the rate-limiting step. Very often, this is the first unique or committed step in the pathway.
Control the entry point.
Exactly. Control the entry point, or the main bottleneck. That's usually where you get the most efficient and impactful control over the whole flow of molecules through the pathway. Feedback and feed-forward regulation.
I've heard terms like feedback and feed-forward regulation used. What's the essential difference between those two in controlling pathways?
Good distinction. In feedback regulation, or feedback inhibition usually, the end product of a metabolic pathway actually controls its own rate of synthesis. It feeds back to inhibit an enzyme earlier in the pathway, typically that rate-limiting enzyme we just talked about.
So like a thermostat, if the cell gets warm enough, has enough product, it tells the furnace, the pathway, to shut off for a while.
That's a great analogy, exactly. So if the cell has accumulated enough of the final product, that product molecule itself often acts as an allosteric inhibitor of the pathway's first committed enzyme. It slows down production.
Makes sense. Conserves resources.
Absolutely. And we saw an example with A and R using ATP. When ATP levels drop and AMP levels rise, that high AMP acts as a feedback activator for pathways that make ATP, like glycolysis, phosphofructokinase 1, and glycogen breakdown, glycogen phosphorylase. It's signaling we need more energy.
Right. So feedback can be inhibition or activation.
Correct. Now, feed-forward regulation is kind of the opposite logic. Here, the substrate, or an early precursor molecule in the pathway, actually stimulates or speeds up an enzyme later in the pathway.
So the arrival of raw materials signals, get ready, more is coming.
Precisely. This is very common in pathways designed for disposal or rapid processing of a potentially harmful substance, or just ensuring later steps keep up. For instance, the urea cycle in the liver converts toxic ammonia into less harmful urea for excretion. This pathway actually speeds up when more ammonia, or its precursor arginine, is available. The substrate pushes the pathway forward.
Got it. Deal with the incoming load faster.
Exactly. And thinking about Ann R again, her low caloric intake means her fuel storage pathways, like glycogen synthesis, probably haven't received the necessary feed-forward signals, like high levels of glucose 6-phosphate, needed to really replenish her glycogen stores adequately. This likely contributes to her fatigue during exercise.
So the lack of feed-forward contributes to her problem.
Potentially, yes. And remember, beyond these rapid allosteric or covalent controls, there's also that slower regulation of the amount of enzyme protein, its synthesis or degradation. This provides a longer-term form of feed-forward or feedback. Like with Al-M, chronic alcohol use leads to increased synthesis of the MEOS enzymes, a long-term adaptation, a feed-forward response to the consistent substrate load, but one with potential downsides. Tissue isozymes and compartmentation.
We talked earlier about hexokinase and the glucokinase, different versions in different tissues. That sounds like another layer of pathway regulation, right? Tissue-specific tuning?
Exactly, it's a crucial concept. Tissue isozymes. Different tissues in your body have genuinely different metabolic needs and roles. So key regulatory enzymes often exist as these tissue-specific isozymes. They are slightly different versions of the same enzyme, encoded by different genes, but they catalyze the same basic reaction. However, they often have distinct kinetic properties, like different chirems or different regulatory responses.
Like the hexokinase, I'm in red blood cells versus glucokinase in the liver.
Perfect example. Hexokinase ensures constant glucose uptake for the red blood cell's basic needs, regardless of blood sugar fluctuations, because of its low cannabin. Glucokinase in the liver, with its high cannabin, only kicks in significantly when glucose is abundant, prioritizing storage. These subtle but critical differences allow organs to specialize their metabolic roles while maintaining overall balance in the body.
Very elegant. And what about just physically putting enzymes in specific places within the cell? Does that help regulate things?
Absolutely, that's compartmentation. It's a brilliant fundamental organizational strategy in eukaryotic cells. Enzymes that work together in a common pathway are often physically grouped together within specific cellular compartments or organelles.
Like the mitochondria for energy production.
Exactly. All the enzymes of the TCA cycle and oxidative phosphorylation are packed into the mitochondria. Enzymes for fatty acid synthesis are in the cytoplasm. Lysosomal enzymes are kept safely inside lysosomes, or sometimes enzymes are assembled into large, stable, multi-enzyme complexes, even without a membrane barrier. Like that MEOS system for alcohol is embedded in a specific cell membrane.
And what's the advantage of keeping them together like that?
Several advantages. It efficiently channels substrates directly from one enzyme to the next, minimizing diffusion time and preventing the loss or unwanted side reactions of intermediate products. It also allows the cell to maintain different chemical environments, like different pH or ion concentrations within different compartments, optimizing conditions for specific enzyme sets. It adds another layer of incredibly efficient and precise regulation. Ultimately, when you look at texts like Marks, you see human metabolic regulation is this incredibly complex, multi-layered system, integrating all these strategies: kinetics, inhibition, allostery, covalent modification, isozymes, compartmentation to ensure balance, efficiency, and adaptability. It's quite the intricate dance. Hashtag tag tag outro.
So wrapping this up, what does all this mean for you listening? We've taken quite a deep dive into the really fascinating world of enzyme regulation. We've seen everything from subtle shifts based simply on how much raw material, how much substrate is available.
Yeah, the basic Michaelis Menten stuff.
Right, all the way to these dramatic shape changes triggered by remote control molecules, the allosteric effectors.
And those phosphorylation cascades acting like molecular switches.
Those protein partnerships. And even the sort of irreversible activation by cutting the enzyme proteolytic cleavage.
What's truly amazing, I think, is just how many sophisticated, interconnected mechanisms your body employs. It's not just one trick. It's ensuring that thousands upon thousands of these enzyme-catalyzed reactions are coordinated almost perfectly. It's this continuous, elegant, biological dance.
Yeah.
Always adapting to your diet, your activity level, your physiological needs at that very moment. It's really a testament to the sheer complexity and elegance of biochemistry, keeping things efficient, preventing cellular chaos.
It really is. And thinking about those clinical cases, LM and NR, we can really appreciate that when these finely tuned systems get disrupted, whether it's by chronic alcohol use or maybe just inadequate fuel.
Yeah, the consequences can be significant. Understanding the regulation helps understand the pathology.
Absolutely. It is truly a marvel. Makes you appreciate the intelligence just embedded in our cells. So, thinking about all this, maybe a final thought for our listeners. What stands out to you as maybe the most surprising or ingenious way enzymes are controlled?
That's tough. Maybe the sheer amplification power of those phosphorylation cascades. How a tiny hormonal whisper becomes a metabolic shout.
Yeah, that's pretty impressive.
And thinking forward, how could really deeply understanding these mechanisms, maybe at an even finer level, open doors for future medical breakthroughs, perhaps new drug targets, or even just better strategies for our own personal well-being and health? Something to chew on.
Definitely something to think about.
Well, thank you for joining us on this exploration of the invisible but absolutely essential forces that govern our biology. Keep digging deeper.
And we'll be here next time for another deep dive.